The Latvian Biomedical Research and Study Center is the leading research institution in molecular biology, biomedicine, and biotechnology in Latvia. The Research Institute was founded in 1993, but its true origin is in the nucleic acids chemistry group organized by Professor Elmers Grens 50 years ago.
In the
60s, a group of enthusiastic young people decided to accept the challenge of studying molecular biology, starting from scratch, without experience and without material support.
Bez pieredzes, bez arī materiāla atbalsta, un rezultātā.
As a result, by the 1970s, we gained recognition in the scientific community and published our first scientific papers in world-class scientific journals, paving the way for biomedical researchers. We moved from fundamental science to the practical application of scientific knowledge in the biomedical field and in recombinant biotechnology.
Pārgājām uz praktiski pielietojumu zinātni tieši biomedicīnas laukā.
In addition to research, the Latvian Biomedical Research and Study Center also serves as a training facility. It is closely linked with the University of Latvia, where many Research Institute staff members teach courses and conduct workshops using the knowledge base and scientific equipment of the Institute.
Each year in the Biomedical Center, some 50 students work in an actual scientific environment on the most advanced scientific equipment to pursue their coursework and their master's, bachelor's, and doctoral theses. We also provide a number of laboratory classes taught by the biology faculty of the University of Latvia. For example, the students visible in the background are performing lab work in biochemistry.
Since its foundation, the Latvian Biomedical Research and Study Center has significantly developed and grown to become one of the largest scientific institutes in Latvia. From the beginning of this century, the Research Institute has established itself as a leader in the field of medicine, developing new lines of research in human genetics, cancer, and other diseases.
B hepatīta vīrusa klonēšana radīja jaunu virzienu tā saucamo vīrusveidīgo daļiņu izpēti ar nolūku radīt jaunus.
The cloning of the hepatitis B virus led to a new line of research in the study of so-called virus-like particles in order to create new vaccines and immunodrugs. Such investigations are important for the development of new drugs, and an important part of this research was conducted here by my professor Pauls Pumpens, my most talented student, who performed extensive studies that were the main focus of our institute.
Pētījums un savulaik šie pētījumi bija galvenais virziens mūsu institūtā.
New lines of research made it necessary to acquire equipment and clinical resources that were not available in Latvia. Over the past decade, thanks to the support of the European Structural Fund and the substantial reconstruction of our research infrastructure, the most advanced world-class scientific equipment was purchased to establish laboratories for complex genetic analysis, microscopy, biotechnology, and molecular protein structure determination.
In 2005, we created the Genome Center, thus establishing the basis for the genome database of the Latvian population. Our database includes a variety of samples and clinical information from more than 28,000 people. This database is now the largest biobank resource in Latvia and the second largest resource in the Baltic states as well as Eastern Europe. Because of this biobank and the equipment acquired within the framework of the Structural Fund, we have created cutting-edge facilities that are among the best in Latvia and around the world. These facilities have given us the opportunity to interact with many foreign groups and perform high-quality studies in human disease research that link disease with genetic factors.
To explore a variety of diseases, especially cancer, and to create new diagnostic methods in modern drugs, it is necessary to study live human cells and use laboratory animals. Such studies require specially equipped laboratories that were unavailable in Latvia until the creation of the Biomedical Technology Complex.
Establishment of the complex began in 2012 within a project of the European Structural Funds. Foreign design professionals were involved, given the unprecedented nature of this project in Latvia. The BMC employees worked together with the design professionals to implement the Biotechnology Complex project.
The building consists of two independent sections, cell biology and laboratory animal blocks, that were placed into operation in the autumn of 2015.
The premises of the research center must comply with the highest standards, which means compliance with specific requirements. For example, the rooms must not have windows to the outside, and uniform illumination and microclimate conditions must be provided.
The clean rooms in both blocks are designed using special materials, including metal wall panels painted with special paint, metal ceiling tiles, glued double glass within an inner frame, hermetic metal doors, and specific floor coverings. All surfaces are perfectly smooth. Lighting, power supply, and other engineering service elements are embedded into the walls and ceilings.
Making the rooms hermetic was a particular challenge during construction. It was also important to ensure overpressure in the rooms in relation to the surrounding spaces. The insulating system provides each room with individual air inflow, and outgoing air is filtered to prevent various infectious agents and aromas from escaping into the environment.
The ventilation system consists of 4 separate ventilation installations that are operated and controlled by a special computer.
The ventilation complex occupies as much space as the laboratories and provides a complete air exchange in each clean room every 5 minutes.
However, the greatest threat to experiments is man. Therefore, these laboratory rooms are accessible only to trained scientists, who must enter through specially arranged changing rooms, where the employee must change clothes and, in many cases, even take a shower.
The cell culture block is designed for working with a wide variety of cells, including patient-derived and virus-infected cells. Any human tissue samples can be a source of bacteria and viruses that can potentially endanger the researcher as well as contaminate other cells in the laboratory.
The cell culture facility consists of 3 separated rooms. In the first room, particularly clean mammalian cells are grown for a variety of experiments. The second room is reserved for cells derived from human tumor tissue, and the third room is intended for work with viruses.
The study of cells can provide clues as to why they have become malignant and how they acquire resistance to drugs. This knowledge is necessary for the development of new anti-cancer drugs and treatment techniques. Cell cultures specially generated in the laboratory are the basis for the development of drugs, as all newly identified drugs are initially tested on cell cultures, and only the most promising compounds are further tested on animals. However, to gain a better understanding of how tumors behave in the human body, to differentiate between tumors in individual patients, and to search for the best drugs for each individual, scientists have learned to generate cell cultures from tissue samples obtained from patients and create complex tissue-like structures.
To comprehensively analyze expensive cell cultures, confocal and fluorescence microscopy rooms and an analytical chemistry laboratory equipped with the most advanced cellular and analysis equipment were constructed next to the clean rooms. The cells are placed in special containers with liquid nitrogen for long-term storage. In these laboratories, it will be possible to develop medicines that are similar to the famous Latvian medicine Riga.
No matter how complex a cell culture system is, it still cannot model how a disease develops in the body.
To understand how cancer spreads in the body and invades the bones, lungs, liver, or brain, and how the immune system fights it, it is necessary to use animal models.
Cutting-edge, world-class research with animals occurs in an environment that is completely free of microorganisms that could interfere with the health of laboratory animals and experimental outcomes, and we define such rooms as specifically pathogen-free.
Ensuring these conditions is a great challenge for laboratory animal facilities around the world. In Latvia, experiments on animals are conducted at a number of scientific institutions. However, experiments in specific pathogen-free environments cannot be ensured at other facilities due to technical limitations.
This laboratory animal facility was designed according to the highest standards, beginning at the project development stage, and it is suitable for studies on mice, rats, and rabbits.
Animals enter the laboratory animal facility through a specialized entry and spend a few weeks in quarantine. Scientists can enter the specific pathogen-free area only by going through an air shower, where all dust particles are blown off laboratory clothing. The animal facility has a number of rooms designated for experimental work with mice, rats, and immunodeficient animals, and for work with infectious models for vaccine development.
One of the major goals of the Biomedical Center is the development of virus-like particle-based vaccines. We now have the opportunity to test vaccine candidates in animal models at the appropriate biosafety level in fully equipped and certified premises and appropriate facilities.
Both minor procedures and surgeries can be performed in each of these complexes. Specific equipment, including cage washing machines and autoclaves, can be used to ensure cleaning and disinfection. Moreover, to ensure environmental control, the animals are kept in individually ventilated cages, and their transfer within the facility is conducted using specific equipment. In the laboratory animal block, it is possible to work with mice that have been bred to specifically study different diseases, as well as mice that contain human cancer tissues. Currently, this is the most advanced research method for better understanding of the biological characteristics of cancer and how it responds to treatment and acquires resistance to drugs.
It is feasible that in the near future, animals with human tumor slices implanted under the skin could be used for so-called personalized medicine. In this way, the animals can help us determine which of the available medicines would be effective for a particular patient, thus making a significant contribution to patient treatment.
The established research complex is a solid starting point for the further development of our institute. I am now standing in the premises where many new laboratories will be constructed. First, the Latvian Genome Center will move here and become a reference point in the establishment of the Latvian Biobank network. A number of new diagnostic laboratories will also be set up. Another major project establishes a cellular product manufacturing complex that will produce drugs that could be used in clinical trials and human treatment. Finally, the new building will be connected to our existing building to facilitate access to various experiments for our scientists and all other Latvian scientists.